Hi everyone, this is our lecture on Hardy-Weinberg population genetics. So let's get started. Why is Hardy-Weinberg population genetics important? Well, let's consider this flash case. A couple is receiving counseling about the risk of genetic disease in their first pregnancy. Although the family history is negative, they are concerned about the risk of phenylketonuria, or PKU, since one of their friends recently had a baby who was screened and diagnosed with the disease. With one child in 10,000 in the United States born with PKU, they want to know the chance that they are carriers. Our learning objectives for this lecture are to recognize Hardy-Weinberg equilibrium as a tool for interpreting genetic variation in populations, to name the assumptions included in a population in Hardy-Weinberg equilibrium, and to utilize the Hardy-Weinberg equation to determine frequency of alleles and frequency of genotypes. So what is population genetics? Well, it's the study of genetics within a population, which is important because it helps us to identify variations within a population and to be able to recognize differences in prevalence of a disease from population to population. Now, we all know that the majority of populations have a lot of changes that occur. For example, how many times have you moved to a new location in your life? People come and go. But in an idealized population, which is what we assume when we apply Hardy-Weinberg law, we can say that no mutation is occurring at the gene locus, that natural selection is not occurring, that there is completely random mating, no net migration, and a large population. Okay, so let's start getting oriented. Remember, we're in the genetics section, so more often than not, we use letters for alleles. Usually, we use a capital letter for a dominant allele, like a capital A, and a lowercase letter for a recessive allele, so a lowercase a. So when we are trying to use shorthand for a homozygous dominant genotype, we usually abbreviate it as two capital A's. When we are indicating a homozygous recessive genotype, we write two lowercase a's. And when we are discussing a heterozygous genotype with one dominant allele and one recessive allele, we write it as one capital A and one lowercase a. Again, this is often the standard notation. But please be careful when reading vignettes and descriptions. I've seen test questions get really tricky and try to flip what an uppercase and lowercase letter stand for. I've also seen question writers use a shape or an icon like a star to indicate an allele. So just like when you're reading a graph and you look at both the X and Y axes labels, make sure before you start doing genetics calculations that you know what the right symbols are for the alleles. What do we have here? It's a Punnett square. So let's say we are crossing or breeding two heterozygotes. So their genotype would be big A little a and big A little a. So let's put a big A here on top and a little a here, and a big A here on the left and a little a here. When we cross, we'll have a big purple A from the top in this first box and a big pink A from the left. And in the box below it, we'll have a big purple A from the top and a little green A from the left. Now in this top right box, we'll have the big pink A from the left and a little blue A from the top. And finally in this bottom box, we'll have a little green A from the left and a little blue A from the top. So this slide is the same as the last one, but we're going to switch from big A's and little a's to Hardy-Weinberg lingo, which uses P's and Q's. So we're going to put a P where all the big A's are, like this, and a Q where all the little a's are, like this. Now when we cross them, we're actually multiplying. So when we multiply a P and a Q together, we get PQ. So we'll have that in the upper right and lower left, where we had a big A and a little a. When we multiply two big A's together, or two P's, we get PP, or P squared. And when we multiply two little a's together, or two q's, we get QQ, or Q squared. Okay, now that we have that all straight in our heads, if P and Q represent the frequencies of alleles big A and little a respectively in a population, then P plus Q should equal 1. So for example, if the frequency of big A in the population is 80% or 0.8, then the frequency of little a in the population, if it were going to add up to 1, would have to be 20% or 0.2. But remember, that's just for one allele. If we want to consider a genotype, then one allele is typically inherited from the father and one from the mother, so there are 2 alleles. For a homozygous dominant, or 2 big A's, then the frequency, like we said on the last slide, would be what? Yep, it would be P squared, or PP, the frequency of homozygosity for allele A. Similarly, the frequency of homozygosity for an allele little a would be Q squared. Now, for heterozygosity, also known as the carrier frequency if we're talking about an autosomal recessive disease, then we need to look where we have a big A and a little a. And this happens twice in the Punnett square as a P multiplied by a Q, so it would be 2PQ. So, if we were to add up all the homozygous dominant, homozygous recessive, and heterozygous frequencies together, then we would have P squared plus 2PQ plus Q squared is equal to 1, or 100% of the population. And this is the Hardy-Weinberg equation. If you have that all straight in your mind, we should be okay. A special consideration is with X-linked recessive diseases. In males, they only have one X chromosome, and if it's recessive, then we're talking about a little a with the frequency Q. So the frequency is just Q. In females, since they have 2 X chromosomes, that would be 2 little a's, or 2 Q's, so Q squared. Now, if a population is in Hardy-Weinberg equilibrium, it means that the values of P and Q remain constant from generation to generation. That was a lot of math. Let's do a quick recap. What are the Hardy-Weinberg law assumptions again? Right, the assumptions are that no mutation is occurring at the locus that we're interested in, that natural selection is not occurring, there is completely random mating, no net migration, and a large population. And what is the Hardy-Weinberg equation? Yep, it's P squared plus 2PQ plus Q squared equals 1. Okay, let's apply what we just did. A couple is receiving counseling about the risk of genetic disease in their first pregnancy. Although the family history is negative, they are concerned about the risk of PKU, since one of their friends recently had a baby who was screened and diagnosed with the disease. With one child in 10,000 in the United States born with PKU, they want to know the chance that they are carriers. Okay, what do we need to know first? Well, how is PKU inherited? It is an autosomal recessive disease. So the alleles would be for someone who has the disease would be these 2 little a's. In Hardy-Weinberg, we use Q's, so this would be Q squared. And prevalence of this disease, so someone who has 2 little a's, is 1 in 10,000. That's nice and all, but we were already given that information, and this couple is interested to know the chance that they are carriers. A carrier is big A, little a, and a carrier frequency would be 2PQ. So how are we going to solve for 2PQ? All we know is Q squared. Well, we can get Q from Q squared by taking the square root of both sides. And then we'll have Q equals 1 over 100. Cool, now we have Q. How do we get P? Oh, remember that the frequencies of each allele should add up to 1. So P plus Q equals 1. And we know Q is 1 out of 100, so then that would make P 99 over 100, which is really, really close to being 1. So let's just say it's 1. So then our carrier frequency is 2PQ, or 2 times 1 times 1 over 100, and that equals 2 over 100, or 1 out of 50. So the carrier frequency is 1 out of 50. Well, we've made it to the end. What's the bottom line here? Remember that population genetics is the study of genetic variations within a population to recognize differences in a prevalence of disease. That Hardy-Weinberg law assumptions include no mutation occurring at the locus, that natural selection is not occurring, that there is completely random mating, that there is no net migration, and that there is a large population. And lastly, please remember that the Hardy-Weinberg equation is p squared plus 2pq plus q squared equals 1. Well, that's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.